just to let you know a little bit about me. As you could probably tell, I'm not from Australia. I'm from the US. But, my early connection to conservation and biology started when I was a little kid. My grandfather was like my main guardian growing up, and he was a photographer, professional photographer that worked for the city of Las Vegas, and he took pictures of celebrities. So like one of his biggest claims to fame was he was Elvis Presley's wedding photographer. He took some of the most incredible photos, iconic photos of Marilyn Monroe, Michael Jackson, Muhammad Ali, and I grew up with all these stories of celebrities. But it was in his retirement that he shared his love for wildlife and for nature. And he fell in love with a new subject through his camera, which was landscapes. So being retired and kind of just stuck with me, he would take me along into natural areas, and he would be really patiently setting up shots, and I'd be out like harassing wildlife, like in this picture. And this isn't a very good picture, but it's my favorite picture he's ever taken because it shows kind of a perfect moment for both of us. So this is me, I'm a little kid, I think I'm 8 years old. And I have a little dip net, and I'm trying to catch tadpoles, and I'm hell-bent on bringing them home and having them like live with me. I just wanted to be around them. And this is how I kind of started learning about wildlife. And this is in the Mojave Desert. So I'm learning about wildlife, I'm learning about how the desert can be very surprising, learning how adaptations occur to allow amphibians to live in these areas. And then my grandfather, also totally at peace in this moment, is just taking a picture. His favorite subject was dead trees, which is random, but he loved taking pictures of dead trees. So yeah, I love this because it captures a perfect moment. So I'm connected with nature, he's connected with nature. We're loving our life. And in hindsight, I didn't really think about it in the moment, but in hindsight, it's perfect. So I obviously continue to be really excited about wildlife, and this is one of the species that kind of captured my imagination. The reason we like these weird animals, herps, it's a strange group of animals in the grand scheme of things to get obsessed with, is because they do crazy things. And gastric brooding frogs are one of the coolest. So females of this species would swallow their fertilized eggs, and their stomach would sort of turn into a uterus. The eggs would hatch, they develop all the way into small frogs, and she'd have the worst case of morning sickness ever. Vomit up the babies, the small little hopping versions of her. And this was really interesting for nerds like me, for documentary filmmakers, for scientists, but also for people that were interested in health and health research. They thought by understanding the mechanisms by which she was able to modulate the function of her stomach to accommodate babies and not digest them, they might be able to come up with therapies for diseases that affect the stomach in humans. So this is all very exciting, but unfortunately this species has gone extinct. So not only did we lose that, that catalyst for imagination and captivating people, bringing them into the natural sciences, but we lost all that valuable information that they could have given us. So what's going on with frogs? So it's not just gastric brooding frogs, but all around the world since the 1970s, scientists were noticing that species were disappearing. And this is happening with really, really common frogs and salamanders. And no one really knew why. And it wasn't until 1997, 98, when a graduate student, Lee Berger, who was working at CSIRO here in Australia, discovered that there was a fungus living in the skin of these frogs that were dying from not just in Australia, but all over. And that fungus is called Batrachochytrium, which is called chytrid. I use it interchangeably for the disease and the pathogen, just to make it simple. It's an aquatic fungal pathogen, and it affects all the major groups of amphibians. That's frogs and toads, salamanders and newts, and then caecilians. I love that I'm in a herp society because you might actually know what caecilians are. They're this really bizarre group of limbless, maybe mostly terrestrial or aquatic amphibians, like worms, but it affects all of them. It's a pathogen that causes a skin infection. So it's two main stages, the motile zoospore. This spore is chemoattracted, so it can sort of smell amphibian skin, the keratin in it, and it swims towards it. It attaches to it through sort of like adhesive glue, and then it produces proteolytic enzymes and then drills with its cell deeper into the skin. And as it matures, it produces more spores. The reproductive structures called zoosporangia start releasing more spores through these tubes. Those go into the environment. They can reinfect the same frog or obviously infect another frog. And just to give you a little perspective, this might be hard to see, but this is skin I took off of a dying frog that we caught a week ago. So we're out in the field. We found a very sick frog. And I just pulled off a piece of skin to culture chytrid from it. And these little dots, these are the spores. This is a 1 millimeter piece of skin, and these are tiny spores. So this sick individual, I just took a tiny piece of a biopsy, put it on a petri dish, and there are dozens and dozens of spores swimming out of it. So that's what they look like sort of in real life. And maybe I'm stuck here. And this is the zoosporangia, how they look under a microscope. So within the skin, these would be underneath the skin layer, releasing spores. So this is a sort of newish problem. So it's been several decades, but it is caused by a fungus that is invasive. So as far as we know, it came into Australia in 1978 once through the port of Brisbane, and the same clone has subsequently spread throughout the whole country. And we think the source is the Korean Peninsula, Southeast Asia, because what has been found in previous research is the genetics of all of the strains of chytrid in the world are represented there. So you can think of it as like the mother of all genetics for the world. So the most likely explanation is that is the source. And another great line of evidence is the amphibians there seem to be unaffected. And looking at genetics, it was also shown that the likely spread of it is through the global trade of amphibians. So amphibians were traded for food. They also were traded around the world and put into all sorts of hospitals as an early pregnancy test. And that was done by, you would have like African clawed frogs as laboratory model frog. Someone who thought they might be pregnant, they would come in, they would give a urine sample, they would inject the frog with that urine, and if the next day it laid eggs, they'd get congratulations. But those frogs are known carriers of chytrid, so they're not affected, but they can carry the fungus. And an obvious mechanism is the pet trade, because there are amphibians traded from the Korean Peninsula that I've seen in pet stores all over the world. So I've been studying this it's been 12 years, and chytrid's terrible, but it's also very clever. And there's some things I wanted to talk about that I think make it a really unique organism. And you have to respect it. It's a generalist. So it's spread worldwide, and it's been highly successful because it can infect hundreds of different amphibians. So there are specific pathogens that will affect one type of host, but chytrid seems fairly able to infect any type of amphibian. So another really cool thing it does is early in infection, a really easy way to get rid of a skin infection is just to shed the outer layer of skin. Amphibians are very good at shedding their skin. They do it very often. But what chytrid does is it produces an unknown molecule that tells the cells that chill out, it's fine, everything's good. And it grows and grows and grows until it does the opposite. And it's like everything is not great, and it just goes full blast. And it does things like destroy the immune response. So it produces other compounds which we don't know. If you want to do a PhD, let me know, because no one knows this stuff. It produces like molecules that destroy the adaptive immune system. So white blood cells that come in contact with chytrid are destroyed. And in very sick individuals, they have metabolic and immune dysregulation. So their immune system goes haywire, it causes a lot of internal damage, and their metabolism completely malfunctions. But not all amphibians respond the same. Like I said, in the native range, the amphibians seem to totally get along with chytrid. They exist in a sort of non-antagonistic relationship, which is on their skin in very low levels, and there doesn't seem to be this really nasty pathogenic relationship. That's the case here in Australia. So even though chytrid's new, there are species here that can handle it. And that's the case for the common eastern froglet, Crinia zignifera. You probably hear these everywhere, even through winter. They can carry it, they survive between years, and what that tells us is they're a competent vector, so they can carry chytrid. And this is bad because they can carry it into places where there are highly susceptible species like southern corroboree frogs. And southern corroboree frogs, they're one of the most susceptible species in Australia. They're critically endangered, and they're heavily reliant on captive management or management in general for their survival. So, to kind of put it into more perspective and to bum you out more, this is not just your average everyday invasive species that causes a little bit of population decline. It's a worldwide pandemic, and it's caused more extinctions than any other invasive species or disease on the planet. So you think of like notorious invaders like cats and rats and cane toads, chytrid still is more devastating, even though it affects only one group. And that kind of puts us in a conundrum. So a lot of chytrid declines happen in national parks, so you have perfectly suitable habitat for the species to live, but once chytrid arrives, it's virtually impossible to get rid of, and you can't just release the frogs back into the landscape. So one of the main ways to keep a species from going extinct, if they're on that trajectory with chytrid, is you simply bring them into captivity, you breed them, and the standard next step is to release them back in the wild. But if there's chytrid in the wild, it's sort of a frivolous process because you're raising a susceptible frog. You can release healthy frogs, but you haven't abated the threat in nature. So that threat, chytrid, will still be there. We know that chytrid tends to stay virulent even if it's been on the landscape for a long time. So nothing has changed. So it's kind of an insane thought to think, oh, we'll just throw frogs at it and eventually we'll figure it out. They may not actually work. So we need ways to conserve frogs in nature, and that's kind of the main gist of the themes of my research. So I have been working for the past several years on ways in which we can allow the frogs to exist in nature and coexist with chytrid. And that is because chytrid's not going anywhere. We have to acknowledge that it's part of the ecology now, and we need to interrupt that relationship or promote the frogs to be more resilient. And I do that for a variety of ways. The main ones I'll talk today about are habitat refugia, our frog saunas, vaccinations and treatment, and then I'll briefly talk about gene editing. The species I work on most is the green and golden bell frog, whatever it's called, aria now, and Renoideae, I think is the new name. It is awesome. It is one of my favorite animals on the planet. It's proudly Australian. It permanently wears the sporting kit of green and gold. And it's the biggest, baddest frog in the ecosystem. They eat everything, including each other. You could think of them as like a keystone frog. So anything they can fit in their mouth is dinner. And I like to think of them as like this tiny version of the Yellowstone wolves, just out there just crunching down all of the marsh frogs. But they're highly susceptible to chytrid. And we know since chytrid arrived, they've gone extinct from about 90% of their populations. And this has been a really sharp trajectory coincident with chytrid. They're highly susceptible in the lab, so it's probably one of the primary reasons. But their susceptibility modulates with time, and that's because of things like temporal refugia. So chytrid likes to be cold, and it kills the frogs during cold seasons. But if you have hot habitat, you can actually keep them on the landscape. So I'm going to talk about two types of refugia that have already kind of been discussed in the literature before we started working on it, and then talk about that in the context of green and golden bell frogs. So the basis for our research was based off of this work done by Rob Wischendorf. And what he found is these frogs from the north of Australia that existed in areas that had these big granite boulders. And what happened is during the day, they would absorb energy from the sun, and during night, they would release radiation. And the frogs would go out and they'd be hunting and basking on them, and they would heat up their bodies. And he took these amazing pictures, where he saw that their bodies were heating up to high temperatures, and this was probably the reason that they were surviving. they use this information of natural habitat or refuge to do a translocation of frogs from an area where they were declining to an area where they had this habitat but they'd never existed before, and it was highly successful. And it was the first use of this sort of refuge idea in conservation. And as I said, a refuge can also be time. So in the case of great and golden bell frogs, which I jumped ahead on a little bit, they have times during the year where they're more susceptible. Again, chytrid likes to be cold, it suffers in higher temperatures, and in summer, the pathogen more or less goes away. And in winter, there's a spike in prevalence. In one of our populations that we work in, it's 100%, so every frog we capture at the beginning of winter is infected. And you have mass die-offs. So right now we're going out and capturing dead and dying frogs. And this happens every winter. You're losing large chunks of the population. They're failing to recruit new members in the population, and it's a huge challenge. But this provides an opportunity. What if we could just keep them warm year-round? So provide that opportunity, that habitat, or that temporal, or like just get rid of the temporal thing, just keep them warm the whole time. Maybe this could prevent them from dying in those yearly epidemics. So this is Shannon Kaiser. He was a master's student. He's now a PhD student at Macquarie. And he did a lot of this early work looking at developing a prototype for an artificial refuge, which eventually we call a frog sauna. Just a pile of black masonry bricks. We put out in the sun. We trialed a bunch of different designs, and we put some data loggers inside. And this is just a graph showing like 1 day of temperature data from these like early pilots. The red line is the higher level of the bricks, the blue line is lower level of the bricks, and then our control is like under a piece of timber on the ground. And that red line is like the upper thermal limit of chytrid. This is where chytrid really starts to struggle to grow. Beyond this, it's fairly illegal, the temperatures. And what we found is that it pretty often, and for a long time during the day, the temperature in those brick piles was getting warm enough that chytrid would have a hard time growing. And this was great. So we could create an artificial structure that can get warm enough in winter, under the winter sun, that we could provide a thermal environment that's not very great for chytrid. So at the same time, I started talking to this postdoc. She's now an assistant professor, Erin Sauer, about how we might test this more experimentally. And this involved using her design of thermal gradients. Because one of the questions we had was, okay, we can make these hot shelters, but what if the frogs just don't want to be hot? This is basic research, but it's for an applied purpose. So we took her design, we modified it a bit because we have a different access to stuff here in Australia, but these are essentially just fence post covers. They're about 1.5 meters long. We had an end that had ice packs in them, and we had another end that had like heat tape that you use for like steak racks or something. And this created a gradient between like 38 down to 8 degrees. We chucked the frogs inside, covered them, and then every few hours or so we would take the temperature with the thermal IR gun. And what we found is this is the temperature for about 70 frogs. This is the upper thermal limit for chytrid. We found that our frogs like to be hot, and that was great data. I remember this is during COVID, and just like leaving and just super excited to share this with anyone. I think harassing anyone about, oh my God, the frogs were this temperature today. And, but it was like basic research. This is like one little piece of information that we need to drive the rest of the research question. And their average is about 29 degrees. So the upper thermal limit is 28 for chytrid. The frogs on average like to be 29. You see there's lots of variation, but every single frog was detected at a high temperature, even though there's lots of variability. So we wanted to test this idea now like experimentally. So Macquarie has these amazing outdoor facilities in our fauna park, and we had just set these mesocosms up. They're these big aquaculture tanks, and we wanted to see if we could test the idea of an artificial shelter in a wild setting. So we did it here. We set each of these up to mimic a pond's edge. So bell frogs in winter, they like to shelter in like the edges of the pond and like a bit of vegetation or rubble, rocks and stuff. So we attempted to mimic that in all of our mesocosms, and we built a hot shelter. So it was a pile of bricks with a greenhouse over the top. And then we had the exact same structure as our control, which was just shaded with a bit of 90% shade cloth. And big shout out to Terry Shine for sewing these for me. That was very helpful. And then in each of these, so we have our unshaded treatment and our shaded treatment. We put frogs. And again, this is during COVID. This is Shannon Kaiser again. I don't know who that is. And we put 10 infected frogs and 10 uninfected frogs in each. And because it was COVID, only Rick was allowed to help me for the whole experiment, so I had probably the most overqualified research assistant for about 4 months. And it was actually a great time because we had nothing else to do. We could just go out, catch frogs, and swab them. And one of the basic questions, and this is really important, is do they actually use the shelters? Fortunately, the answer is yes. And 74% of the time that we went and sampled our frogs, they were inside these brick piles. Does it actually make them hotter? I hope so. This is a graph of body temperatures that we took of the frogs as we were capturing them. And those in the red line are from our unshaded, so our hot treatments, and those in the blue are our shaded, sort of colder treatment. And they're about 3 degrees warmer in the unshaded structures. It doesn't seem like a big difference, but for chytrid, that could be the difference between all of them dying and all of them surviving. There's a really fine edge that can tip the scale in favor of the frogs. And that's what we're finding here in terms of infection. So this graph is of infection intensity over the course of the experiment. The y-axis is like how much chytrid is on their skin. The x-axis is weak. And the red line is our hot treatment. And you can see over time, they're carrying much lower infections. And this is log scale, so these are like 100, 1,000 times different on average. So from this, it looks like if we provide them with these shelters, they'll heat up, they'll carry lower infections, they do clear them much faster. But we're also interested in if they were able to survive these infections, what happened to their long-term resistance. Amphibians have remarkably similar immune function to humans. They have an adaptive immune system. They can be vaccinated. So my question was like, are they vaccinated now that they've been cured? So this was a separate study we ran just looking at their resistance in the lab. This is a graph of survival. So the black line is those that had chytrid infections that were cleared with heat. The red line is those that were just getting a heat treatment. They never encountered chytrid. And then both groups were challenged with chytrid at the same exact time. So one group, the black one, you could think of them as vaccinated, and the red one, you could think of them as not vaccinated. And what we found is there's about a 23 times greater chance of survival if they survived that first infection. So 86% in the vaccinated and only 22% in the heat control. And this to me tells me we have a really cool story. So we build something, it's cheap, costs $1 a brick. The greenhouse is a little bit pricier now, $40. I think the original ones we got from Aldi. You could cheaply make them. The frogs use them, they heat up, and they can gain resistance. So it's kind of like this little propagation engine. So the frogs use them, they get better, and they're leaving healthy and also more resilient to future challenges. So there was this lovely filmmaking student that came a couple months ago, and she wanted to tell this story really comprehensively. So she provided this information, or this video, and I'll walk you through it. So how they work, a frog's finding, like I said, they heat their bodies up, and they cure their infections. And even if they are living their lives and they don't come in contact with one of these structures, they're gonna have resistance to chytrid. So they'll be in the environment, this one might be picking up spores again, but that second infection is gonna be really mild. They're gonna be able to get rid of it and survive. So we're propagating resistance. And this was a huge innovation for chytrid. It's been over 20 years since it was described. And I got a lot of press, but one of my favorite things is I got the word frog sauna in the New York Times. And it was the first time my parents were like, oh, are you smart? we got to work right away. So the Australian Academy of Sciences and Margaret Middleton Fund helped us out with some funding to field trials at Sydney Olympic Park, and we have those trials ongoing this year. So, I've talked to you about our frog sauna. The next thing I'm going to talk to you about is vaccines. Does anyone know what's going on in this picture? Does anyone old enough?
The polio vaccine.
Yeah, exactly. So the polio vaccine. Does anyone know what the polio vaccine is?
Sugar.
Is it a weakened virus?
Exactly. So a weakened version of the polio virus. So when I was first studying chytrid, I was doing it in Las Vegas, at the University of Las Vegas. Chytrid is there as well. And I remember seeing this in my undergraduate immunology class. They're using live versions of the pathogen as a vaccine. And indeed, the very first vaccine, Edward Jenner and the smallpox, the milkmaids don't get smallpox idea, they were using weakened or different versions of pathogen to inoculate people against something much deadlier. And I just finished up a study and wrote a paper about how these endangered frogs probably didn't die from chytrid because when we gave them chytrid in the lab, they didn't die. But then I isolated chytrid from the local environment, did the same study, and I was like, oh, just kidding. they're very susceptible. It just depends on the strain. And I ran into my professor's office, and I was like, why don't we use this weak one as a vaccine? And he was like, all right. And because at the time, no one thought that you could vaccinate the frogs against chytrid. Like I said, it paralyzes the adaptive immune system. But we knew when we gave it to our frogs, they were able to get rid of it themselves, this weakened version of chytrid. So I gave it a go. And that's the data from, I think it was 2021 when this paper was published. This blue line is infection load of the frogs that were given the weakened version of chytrid, so the vaccinated frogs. And they do get infected. None of them die, but they get rid of their infections, or they very closely, or they very significantly reduce them to very levels that aren't dangerous for them anymore. And then when we challenge them with something incredibly nasty, and then we challenge a group of frogs that had never received the inoculation, what we find is very obviously a vaccine effect. So those in the red line, they haven't been vaccinated, and their infections skyrocket until every single one of them dies. So 100% of the frogs we didn't vaccinate died, and the ones that we gave this low virulence chytrid, the vast majority, 80% of them survived. And this is really cool, because no one in our field thought you could make a vaccine, especially not one with a live version of chytrid. And moving that forward, coming to Australia, I've just repeated that same exact study with bell frogs. We found the same thing. Bell frogs can gain resistance if you use heat. We also can do the same thing with antifungals. And why I like this is you can just capture wild frogs right before the epidemic hits. Like I said, when winter hits, the infections skyrocket. I was like, why don't we just treat them right before winter? And that should have the same effect. And that way we don't have to have captive breeding and all these permits and stuff. We can do it on site. Sydney Olympic Park has on-site facilities. We can go out there, capture the frogs, and treat them. And that's what we've done. So this is my pop-up vaccine clinic that we built at Sydney Olympic Park at the Newington Armory. And I love this because I got my COVID vaccine just down the street from here. And I think it's pretty fitting that now we're doing frog vaccines in this lab. They also have artists in residence all around, and they always like, what are you doing here? Just all these tanks of frogs walking in. I'm like, oh, don't worry, they know who I am. I'm always setting off the alarm. But it's a good time because we get to watch in real time our efforts work. We bring in frogs that are on death's door. We're able to get them better, get them back out in the wild. And that's what we've done in an experiment where we've captured every frog we could catch, treated half of them, and the other half we left untreated, but kept them at pretty nice temperatures so their infections wouldn't go up. So I have solid control. And these are the results from our field trial from last year. We're doing it again this year. These ones in red are those that we didn't treat. And when we go out and capture them 4.5 weeks after housing them indoors, they're infected, their infections grow over time, and then we stop capturing those frogs. Those that we treat, they get reinfected at low rates. This is not surprising that these are lower because chytrid grows incredibly slow on our frogs. So the fact that we capture them and they have low infections is biologically meaningless, really. But what's really cool is when we capture them the next time, they've all lost their infection. So 100% of the frogs were chytrid negative. And this tells me that works better than even in the lab. The plan now is to continue doing this. I think, not that it's, as you know, amphibian populations do this a lot, so whether or not our efforts are making a difference, we won't know for several years. But when we started this, they had the lowest number of bell frogs ever at this particular site, and there's triple that this year. And we're recapturing those frogs all the time that we've treated. So at least we're not doing any harm, but hopefully we're doing good. And the final thing I'll talk about is gene editing, and it's gonna seem like a hard transition. So those other two things, habitat shelters and vaccines, are things that are ready to go. And they work at the scale we're looking at, so in the local populations. We can train managers on the ground to do it. It's something we can hand over. It's a feasible strategy for these systems, but chytrid is a massive problem. And there are species that don't respond to this. You can't heat up a corroboree frog to 30 degrees and expect it to do very well. They're an alpine species. We've tried to vaccinate corroboree frogs, it doesn't work. And they're really kind of out of options. You have species being brought into captivity, a dozen or so in Australia, that probably gonna rely on captive management for survival. And that is not sustainable financially, to continue to bring more and more species into captivity. But also, when you bring a species into captivity over multiple generations, you're selecting for individuals that are good captives. And every generation they become less and less fit to live in the wild. And there are ways to kind of slow this down. So for example, corroboree frogs, an amazing strategy is they built these outdoor exposures that exclude frogs that bring in chytrid, like crinia, and they go bananas inside. They breed, there's tons of frogs, they're exceptional. But if we want frogs in the wild, we have to find a way to make them resilient to chytrid. And to me, this is very controversial, obviously. I know it sounds like a bit crazy, but it may be one of the only strategies we have for a long-term permanent solution. And I'll always be working at this from an experimental perspective and doing it from the most conservative, effective way. But I wanted to present it here because this is who else is going to maybe understand it and maybe have good comments or criticisms of it. And we made some progress. So we wanted to do some like proof of concept, really early stuff, just to see if this is a technology that we could adopt. I'm an ecologist by training, so to see if I'm smart enough to pick up something else. And our first experiment was a proof of concept, just to see if we can knock out any gene. But what was cool is I got to take a foray into completely new fields, like in reproductive biology, and this involved frog IVF. So going out when the frogs are breeding, very rudely interrupting sex, stripping the male off, and then putting them in separate tanks and waiting for the female to start ovulating and shedding eggs, collecting the eggs, fertilizing them, and then using those for microinjections. And these are the results from an experiment where we knocked out a pigment gene, tyrosinase, and the resulting animals would be depigmented or albino. So this is like a 1 day old green and golden bell frog tadpole. And the first place that you see pigment in a tadpole is in its eyes. So if you see like the little black circles, that looks like a perfectly normal tadpole. But here are this is an injected tadpole, and you can see they have a kind of a halo, and there's a lack of pigment. And this is really exciting because it looked like what our experiment had worked. And when we screen tadpoles, it looks like we can do this at really high success. We can do this really effectively. So almost 100%, 97% of our tadpoles that we injected as embryos have depigmentation. And this is what they look like as frogs. So on the left, these are wild type frogs. On the right, these are ones that we produced through microinjection. And what happens with CRISPR is the first generation you make, they are called mosaic. And you can see it really well with like pigment knockouts, because they look like a tile mosaic, but they have splotches of pigment. And, but we actually get individuals that are fully albino too. So this is like telling me that it's really effective. We can get it to work really well. pretty good. And these have just had their first batch of offspring, so this is one of the first tadpoles that came from our parents. And this is that same tadpole as a little frog now. So they're just gold frogs now. So what now? I made some like ugly frogs. That's not really the point, right? The point is chytrid. And the general approach is we're just gonna use this as a new experimental tool to understand chytrid, understand resistance, understand tolerance, and then use those learnings to generate frogs that have resistance. And that could be, if we find genes that are important, it could be something like selective breeding. It doesn't necessarily need to be genetic engineering, but it's a new tool in which we can study this very complicated disease and the immune system, which even in humans is a black box. So moving forward, I think at the very least we have a powerful way to kind of modernize our field, move it into experimental approaches that we already use to benefit human health, agriculture, aquaculture, and use it for good. Thank you.
Questions?
I did my Master of Research on human fungal pathogen. Why hasn't anyone talked about antifungal vaccinations in humans and other animals? Like, if you've already successfully shown results from this weekend fungus strategy.
Yeah, so it's a great, so the question was like, how come no one's talking about it with humans? Well, there's like 3 reasons. Fungi are weird and people just don't want to work on them. There's not a lot of funding for fungal pathogenesis, fungal pathogen research, but there is no approved human fungal vaccine. And when I talked about CRISPR and maybe getting money for like health science, we have a model system where we can vaccinate an animal. The way the fungus infects humans is through mucosal layers, the skin. We have an excellent model to study it. So no one really talks about it because it's just this, there's like this bias against fungi for some reason. At the treatment level, fungi are really close to us. So it's much easier to make an antibacterial antibiotic, or much easier to find an antibiotic because they will specifically target something in a bacterium that's very different from humans. Most fungal treatments are highly toxic. It's almost like chemotherapy because you're targeting things that may be similar to what we have in our own bodies. There's off-target effects. Why there's no human kind of fungal pathology stuff? There is a bit. So Candida auris is like a big hospital infection people pick up, usually when they're immunosuppressed. And it's becoming a big issue. So I just looked at the student poster today at Macquarie, and they're estimating something like 50 million people a year are gonna die from fungal infections by 2050. Right now it's about 3 million. And yeah, we've done it in frogs of all places. So yeah, I think there's a lot of opportunity to use this as not only for conservation, but this frog could be a hero for us too. Yeah, Alex.
Have you done any of has anyone else done the vaccination stuff with other species? To me, you showed it worked in one species, and then you said that in things like the corroboree frog, it didn't really work. Is it being tested more broadly?
Yeah, so the very first experiments all didn't work. So what happened is it was shelved for a decade because everyone's like, it's not gonna work. What we knew about chytrid is it paralyzes lymphocytes and kills them. So the prevailing idea is that there's no way that a chytrid vaccine would work because even if it did, once they get infected, the chytrid was going to destroy their immune function. There's one paper with Cuban tree frogs, which has lots of problems. It's not a declining species. They're not super susceptible. They put it at temperatures they've never experienced in the wild, and they live 11 days longer, which is not a biologically meaningful thing. There's a group in Belgium. They worked on the Moroccan midlife toads, and they found the same thing. Low virulent strains can be protected. And I did it in 2 species in the US, 2 leopard frog species, bell frogs. We'll try it in, I have a few other species, but I will try it in. But yeah, it's not going to work for every species. I think some of them, they die because their immune system kills them. They have immune dysregulation. We know that from some transcriptomic studies, like looking at the immune response from like what the cells are turning on and off, and the genes they're turning on are destructive. So they don't have any benefit to the frog. And there are human diseases that do that too. Think of the Spanish flu. It killed a lot of healthy people because their immune systems were strong and they overreacted and they lungs full of fluid. And whereas like maybe older people that had or people with weakened immune systems, they had a weaker response to the flu and they did better. And we think that might be the case for some so it might be why the vaccine doesn't work. So the immune response that they mount, even if it's an adaptive response, it's destructive. Yeah.
So it seems like you've followed all these different approaches, right? And sometimes the vaccination works, sometimes it doesn't, sometimes the sauna is going to work, sometimes it's not, depending on the sort of like the characteristics of the frog. So you try and sort of develop like an adaptive management sort of approach where, maybe in this situation you take this approach, in this situation you take the other. Is that sort of the goal of your research?
Yeah. I didn't mention it, but I partner really closely with the threatened species officer, and we're working on a captive breeding for release experiment. But we're bringing in frogs and vaccinating them. So right now, considering green and golden bell frogs, the adaptive management plan is to do these things that we've already developed now, and they're supporting the research we're doing for more longer-term solutions. And I have projects with Toronto Zoo, and it's exactly the same thing, looking for more long-term things at the research scale. But then we already have things that work, and we can do it now, and that's going to keep frogs in the landscape. And there are frog species that can eventually adapt. So if we keep them on the landscape long enough, you buy time, they might figure it out on their own. So yeah, it's definitely part of my daily thinking.
So you plan to eventually have like a universal treatment option for like large number of different species, or maybe for specific threatened species like the green gulls and crocodiles?
I mean, the silver bullet would be great. Yeah. I don't think we get there with the vaccine or hotspot shelters because there's thousands, there's 8,000 species of amphibians. It's growing every day. I think it would be a kind of mean to them and a disservice to. It would be, how would I describe this? I wouldn't be appreciating the diversity that exists if I said yes. But if we develop like a genetic solution, it might be as simple as like looking for that gene in species or using gene technologies. But that's more like for species that are really, really out of options. So it's not something I would pull the trigger on for any old species. Green and golden bell frogs are going to exist in the wild for a long time. But crowberry frogs probably won't. So I think it depends. You have to make those decisions based off the case.
In terms of the vaccinations or the immunization efforts of introducing a weakened version or a lower sensitive version of chytrid, when you're re-releasing or planning to re-release those individuals that have had exposure, is there much of a worry about, bell frogs might be great with it, but then another species that's within that area is gonna, you just brought in something that's gonna take them out?
Yeah, you have to make careful decisions. So right now, I want to do this in the field, but we'd be bringing in and weakening a strain that's from the site we're working in. So you're not introducing anything new. Yeah, and you'd have to do that at every site that you want to work on. And even though chytrid is a clone everywhere in Australia, depending on where you take it from, what species you take it from, it can be different in its virulence. You can't just assume it's always going to be the same. So yeah, I'm starting to again isolate chytrid from all these different sites we want to work at, so we can work exactly with what's already there.
So how does the run of falls go that's down in Australia and in Victoria?
Yeah, it's definitely susceptible to chytrid, at least in laboratory trials. But
Is it harder to deal with because it's colder climates?
yeah, I reckon this would be a tougher. Yeah, we have to do the research. The thing is, people send me emails like, "Will this work in this species?" And this, I'm like, I don't know.
So they're so closely related to the Aurea.
Yeah, if I had to bet, I think it would work. But yeah, I for some reason I get emails from consultants all the time. They're just going and doing it, and I'm like, you probably should do the research first to show that it's not causing harm.
Yeah.
What I don't want is my research to be like bastardized and used in a way that actually causes harm to species.
So do they know why species like striped marsh and the crania are less susceptible?
Do you know why? Do we know why?
Yeah, like, you know how you said cream and striped marsh and that can live with the chytrid? So do we know why?
I have no idea. We don't, actually, that's like, there would be a dozen or more labs, not just researchers, labs that work on that question, and we still don't really know. I think the challenge is we need to see people that are immunologists. We need people from outside herpetology and conservation to come in with their expertise and modernize our approaches, or else we're never going to get to that answer, because we have an incredible depth of knowledge on the biology, the ecology, but we don't have the tools yet. So while human immunology is like here, they're using AI to identify specific cell types in a sample, we're still injecting frogs with bean proteins and seeing how swollen their legs get. It's like not the same level of research. So I think, yeah, we need some outside eyes to come and look at it with a fresh perspective to get there, because there's too many people working on it for us not to have made 20 years we haven't made any progress really on that question.
Yeah.
To me, it's frustrating, but we have some candidates. We know there are some genes that probably provide protection, but in order to link a gene to a cause, you have to do reverse genetics. So you have to knock out the gene, and you'll get the resulting phenotype. That's what's done in human
If
models.
not the reason that they survive it, then, yeah.
Yeah, but it wouldn't be for it would just be for research to understand like what exactly was making them resistant or not.
Yeah.
Yeah.
Anyone else? No?
Is there a time period that the frogs have to stay in their little frog sauna for this to be helpful to?
Yeah, I mean they only get warm for a couple hours a day.
Okay.
So a couple hours a day over several weeks is probably enough. I didn't show, we had another experiment where we infected them and put them inside the thermal gradients, and the frogs that could run around it and pick whatever temperature they want, they lost their infections in about 5 days. Whereas the frog that we just kept hot, it took them a couple weeks. So like that ability to thermoregulate is probably super important.
That's interesting.
The frog sauna, I probably missed the point. It is a voluntary thing for the frogs, isn't it?
Yeah. I should have clarified. We're not just shoving them in the bricks and yes, we found them inside.
But they're in the green, they can come and go from the greenhouse if they wanted to.
Yeah, so there's like a little 3, 4, 5 centimeter gap, depending on who sets it up.
And it's all solid in.
Yeah, and we know that they kind of use them, in the wild we set them up in little groups for the sole purpose is we're worried about maybe having a little trap where predators can go in and just eat all the frogs. And we like caught a frog, released it back into the same brick, and then like we went to the next brick pile and that same frog was there. So they used them and they escaped very cleverly between them. So it kind of serves the purpose we hoped it would. It's also a way to confuse predators by having multiple of them. But yeah, they can come and go as they want, and they can do it without us even noticing.
Alrighty, please reserve one more, that was it.
Oh, I did add on.
Yeah, sure.
I'm not sure if this is really within your field or not, but just in terms of, I feel like any of us that are doing field work are trying not to spread chytrid.
Yeah.
Do you have any advice for what we can be doing to not spread it? Should we be spraying our boots down? Should we be drying everything out? Like what works, what doesn't work?
Yeah, the most effective way to kill chytrid is to dry it out. That said, I always spray everything with like F10 bleach. And I have like, if you're working in tons of different sites, obviously it's not possible. Right now I'm only working in 2 sites. I have like specific beats for each site I work at. Because you just need a bit of wet soil to carry chytrid. Don't move frogs around is probably the best thing. Because frogs are like, it's very hard to spread chytrid actually. To me, in my mind, I think it'd be very hard for you to spread chytrid if it wasn't via frog. Because it can't live outside of frogs very long. It dies, will die out in the soil in a few days because it doesn't compete well with other microbes. It really seems to need the frog skin to replicate. So it's not like other fungi that can just live off the dead stuff. So as long as, I think F10 drying out is probably good. And if you can, like have dedicated equipment to certain sites if you're only working in a couple.
Cool, okay, once again, please thank you.